Ms. Patterson here. Today we're going to continue with our macromolecule unit, and we're going to talk specifically today about carbohydrates. So there's 4 macromolecules as a review. We have carbohydrates, we got proteins, we got lipids, and we have nucleic acids. I'm going to do each of these videos individually, so if you want a specific one of these, go back out and find which one you want, and that's the video you can watch. So today we're going to cover carbohydrates. So carbohydrates, they're a real quick source of energy. If you guys are really tired and you're at school, you guys will eat a candy bar or you'll drink a Coke. And that's putting some sugar in. Your brain needs sugar to be able to function. So when you drink that, eat that, it all of a sudden starts to kind of perk you up for a little bit. But if you eat too much, it will kind of bring you down. You have too much in your system. So it's a quick source of energy. You know you have a carbohydrate when you look at it and it's a 1-to-1 ratio. Yeah, I can have different size sugars, but for example, like glucose, it's C6H12O6. I have 1 to 2 to 1. My carbons and my oxygens will be the same number. My hydrogens will be double whatever those are. So I have a 1-to-1 ratio. So carbons, hydrogens will always be the same. Carbons and oxygens will always be the same. Hydrogens will be double. So here I have C3H6O3. That tells me it's a carb. This one here also tells me is a carb. It's going to be a type of pentose sugar. And then here I have glucose, which is, so these are all going to represent a carbohydrate. I can have what's called an isomer of a carbohydrate. So each of these is glucose, C6H12O6, but you can see my structure is a little different. I have an H here, OH here. I have a double bond oxygen here, and I've got double bond oxygens here. So these change. One is glucose, one is fructose, one is galactose. So they're all the same type of sugar, but they all have a different function. To put a carbohydrate together, I have to do what's called polymerization. And this is going to be where I have small individual units, which is my glucose, my small little molecule that represents a carb. That's going to be my monomer. I put them together, I can make very long carbohydrates, which is a polymer. So my monomer is going to be carbohydrates. Okay, my monosaccharides would be my small individual units of a carb. I polymerize them. I'm going to put them in and do some type of synthesis to get them together. And then I make a polymer chain, and this is going to be a long string of monosaccharides, which is a polysaccharide. A polysaccharide makes a long chain of carbohydrates. I can break them down and form them with water. So dehydration reaction, if something is being dehydrated, water is taking out. And so this is how monomers, and carbohydrates are a monomer. So these monosaccharides are sitting there and they want to get bonded together. Water will be pulled out. I'll have an OH from one and H from the other get pulled together. We'll make water. This water leaves, and then that oxygen is going to be covalently bonded between my two monosaccharides. My two carbohydrates will be bonded together. They also call this condensation reaction because I'm condensing to make water. So my formula is A plus B equals AB plus water. And to break apart my monomers, if I want to break apart two monosaccharides, break down a polysaccharide into its monomers, its monosaccharides, I'm going to put water into the system. So here I have my molecule. I take water, I add it in. I'm going to have an OH group over here. Hydrogen's going to come over here. And now I'm going to break down my two monosaccharides into small individual monomers of carbs. My formula for that is AB plus water is going to give me A plus B. I can have a disaccharide. Disaccharides are two monosaccharides put together. A disaccharide, here I have glucose, here I have glucose. I'm going to put them together. Dehydration synthesis, this water is going to get pulled out. And then when it does, I'm going to have them bonded together. This is a covalent bond. And these two glucose molecules make maltose. So now I have a disaccharide, which is a covalently bonded carbohydrate. So that will leave, and I have my bond. We call these a glycosidic bond. Sometimes you hear a glycosidic link, same thing. So carbohydrates have three types of bonds, I guess you could say. You have ones that are going to be kind of simple starches, and these will all have their bonds going in one direction. These kind of coil, but they kind of stay fairly straight. They don't branch out very much. You have ones that are more glycogen. These are going to be same thing. All my bonds go in one direction. And so these kind of like to branch out. And then I have cellulose. When you look here at these bonds, you notice something different. I have one bond this way, one bond's up, another bond's down, another bond's up. These alternate. And this is something very different. With cellulose, it's very straight, very flat. And we see this in plants. We see this as the structure that makes plants stand up. Plants don't have a skeletal system, so they have to have something that's going to be a structural fiber, some type of support system for the plant to come up. So the cellulose kind of fits that bill. This one here is more starch. Plants make that. Glycogen is something that's made by animals, so this gets made by that. So cellulose is made by animals. But when you think about foods that have no calorie benefit, so if I went and I ate celery and all I ate is celery, I would starve. If I went outside and ate grass all day long, I would starve, because the plants have this structure. And I don't have an enzyme or a molecule, protein, that can come in and break these bonds down. It just can't work. I'm not able to do that. Now there are some animals that can. If we look at cows and deer, they have a second or another stomach, a second type of stomach that allows them to be able to have the enzyme, the bacteria needed to be able to break down this bond so they can eat grass. Where here we have the enzymes in our stomach to be able to break down those bonds. So I can break down like potatoes, I could break down types of meats, I can do all that, but there's some plants I can't eat. But we need it. This is kind of when we talk about fiber. This kind of goes in and it allows your intestines to go through your intestines and pick up pieces, pick up material that you can push out and pull waste faster from your system. So we need certain types of fibers. So here I have different monomers. I have different monosaccharides, and they're all put together with a covalent bond. So I've had dehydration synthesis put them together. If you're performing an experiment that involves a hydrolysis reaction, so hydrolysis is when water's coming in to break these bonds. Hydro means water, lyse means to cut. I'm going to hydrolyze these. So the polymer you're working on with is amylose starch. I know it's some type of carb because it's a starch. There are 10 monomers of glucose that make up this polymer. How many water molecules will you need to completely hydrolyze the amylose? So how many water molecules do I need to completely break apart this monomer? So I have 10 pieces and I have 9 bonds. So for me to break each bond, I need 9 water molecules to do that. So I bring in my 9 water molecules. They're going to go in, they are going to hydrolyze those bonds. And now my polysaccharide has now been broken down into individual monosaccharides. Molecular bonds of a disaccharide can change your total outcome. So if I take these two individual monosaccharides, I know there's a carb because they're 1-to-1 ratio, 1-to-1 ratio, and I put them together and I make them to a molecule. I'm not going to have C12H24O12. I'm going to have C12H22O11. And this really gets students wound up, okay? But they're like, but it's not a 1-to-1 ratio. It started out 1-to-1 ratio. But what did I have to remove out of it? When I look at my carbons stayed the same, right? I have 6 here, I have 6 here, so that gives me 12 carbons. I had 12 here and 12 here. I should have 24 carbons, but I'm missing 2. I have 6 oxygen, 6 oxygen. I should have 12, but I only have 1. So how many hydrogens am I missing? 2. How many oxygens am I missing? 1. What makes up water? H2O. Okay. So you can show that by going in there. I pull out my water molecule. So now my hydrogens, I'm going to pull 2 hydrogens. I'm going to pull an oxygen out. So now I have my formula. I'm going to add those up. And I should have this, right? If it's a true carbohydrate, I should be 1-to-1. But I have to remember, I've removed this water molecule. So I no longer have these extra 2 hydrogens, and I don't have this extra 1 oxygen anymore. So now I have 22, and now I have 11. Okay, so my formula ends up being this. So now I have a trisaccharide. So if you want, go ahead and pause it, and you guys can figure out my molecular formula. If I was going to bond these together, what would be my formula? So I'm going to add together my 3 monosaccharides. I take out my water, and I have to figure out what I've got. So I'm going to have 3 sets of carbon. So I'm going to have carbon 18. Hydrogens will be 36. Oxygens will be 8. I now have to remove not just 2 hydrogens, I have to remove 4. And I no longer remove just 1 oxygen, I'm removing 2. So I go in, I remove 4, and now I have 32. I go to my oxygens, I no longer have 18, I have 16. So my formula is C18H32O16. So polysaccharides, these can do a lot of different functions. I can store energy. I can do that with starches and glycogen. Cellulose is going to be some type of structural benefit. So here I have amylose, which is going to be a long line. I have starch, which is going to be kind of branched out, but it's kind of pretty straight. Glycogen is pretty branched. It's going to have a lot of different carbons going off. And cellulose is very straight, very structured. This is also what gives it a lot of strength. So starch, here I see my glucose molecules, my monomers of carbohydrates. My bonds have been put together, water has been taken out, and I have my dehydration synthesis has put my pieces together. I'm going to find this in plants. Most plants are going to have starch, potatoes, corn, breads. All of these things here are going to produce a type of starch, and that's a storage for the plant. Extra granules that don't get used within the chloroplast will be stored within plastids or the meaty part of the vegetable or fruit. Glycogen, this is going to be found in animals. So here I have my, again, each individual monomer. They're all dehydration puts them together. Here I have a branch coming off my carbon. So I have a glycogen branch there. These are going to be found in liver and muscle cells. So here I've got muscle cells. So I'll have glycogen stored in those. Hydrolyzing this is going to release glucose whenever it's needed. So if you've ever done sports, sometimes track, cross country, they'll have like a pasta party the night before, and they're carb loading people so they can have extra energy the next day. The body will store that and then use it the next day as fuel. Cellulose is the last one. This is a structural polysaccharide. This is going to be the tough wall of the plants. So celery, these little hard parts outside, is going to be the cellulose. This is undigestible in humans, but it's good. We need it to be able to scrub our intestines, get rid of certain waste inside there. I'll have it's a different isomer of glucose. Remember, an isomer is when I still have C6H12O6, but my setup is different. So cellulose is an isomer of glucose. I'm going to have glycosidic links are going to be different. Remember, one goes down, one goes up. So one goes down, one goes up. Dehydration synthesis is going to put them together. But I'm going to have a hydrogen up here. And then you notice here my hydrogen's down here. This flips. And so I have a different, so this is kind of why my chain goes up and then down, is because my glucose, my structure is different than my other one. So my bond gets different. Okay, chitin. This is going to be found in exoskeletons of arthropods. So it's going to be a fibrous structure that goes in. It gets really hard. So some arthropods will have to like shed their skin, so their shell will fall off, and then they have to regrow their chitin exterior again. This is going to provide structural support. You also find some in fungi also. Okay, that's an overview of carbohydrates. If you guys have any questions, let me know. Talk to you later. Bye.